Today we'll be going over the Lac Operon, and it's one of my favorite processes because I find it very precise, unique, and very interesting. So I'll help you guys understand how it works, how, why, and all that good stuff. Now, the Lac Operon is a genome found in E. coli. Now, E. coli is found in the intestines of humans and animals. And what E. coli does is break down the foods that we eat. Correct? So now we're going into the specifics. What genes are responsible for this? How does it happen, right? Now, this Lac Operon is specific to lactose. When you drink milk, any dairy product, cheese, whatever you want, I don't know what you guys eat, but any type of dairy product, it has something called lactose. Now, E. coli has many other operons. However, today we'll just be focusing on the Lac Operon. And so what E. coli is able to do is able to transcribe these genes, the LAC-Z, the LAC-Y, and LAC-A. These are the genes responsible for breaking down lactose. So let's go over what each gene does. The LAC-Z, it produces an enzyme called beta-galactosidase. Beta-galactosidase. And what it does is it cleaves that lactose into simpler sugars. Those simpler sugars will be glucose and galactose. Okay? That's what the LAC-Z does. The LAC-Y, it produces a protein that embeds itself on the in the cell membrane to allow lactose to enter the cell. So as you guys know, let's just draw a quick one. I don't know if this is a good drawing, but like this is the cell membrane, and you have these. Let me draw that in a different color. And you have these lactose coming in. So what the LAC-Y does is creates a protein on this cell membrane, like a little hole or something you can think of, to allow this lactose to enter the cell. Okay? Now, the LAC-A is very complex. That's why usually the most important is the LAC-Z and LAC-Y. The LAC-A encodes for B-galactosidase transacetylase. Now, for this video, we are not gonna go into LAC-A, just LAC-Z, LAC-Y, because those are the most important. And just know that these three genes is what help break down lactose into simpler sugars. Okay. Then over here we have the capsite, and what the capsite does is it enhances transcription. And a little later in the video we'll go over what that means. It promotes RNA polymerase, and it acts as a transcriptional activator. The promoter, this is where the RNA polymerase binds to. You know, the promoter is like, hey, RNA polymerase, I'm right here, you know, bind to me. The operator is where the repressor will bind to. Okay, so in this video we're gonna go over 4 different scenarios. Okay, so what happens when there is no lactose present? Okay, there's no lactose. What's going to happen? Well, the genes are not going to be transcribed. Remember, these genes were responsible for breaking down lactose. But if there's no lactose, Why transcribe those genes? You know, it's E. coli, the E. coli genome. It's going to be like, hey, I'm not going to waste my energy transcribing these genes if there's no lactose. What's the point? Come on, I'm not wasting my time. I want to chill, So how is it able to not transcribe these genes? Something called a repressor. And that's what binds to the operator. It's going to bind, say this is the repressor. It's going to bind to the operator. So then, when RNA polymerase comes in, it tries to bind to the promoter and transcribe, but that's not going to happen. It's just going to bounce back because you have this operator in the way. It's saying, hey, RNA, you're not going to transcribe these genes because there's no lactose present. So what the repressor does is it basically is like a blocker, It blocks RNA polymerase. No, you're not going to transcribe these. There's no lactose at all. Don't do it. Don't waste your time. We're not going to do that. Okay? So that's scenario 1. Very simple, very simple. Remember, genes need to get transcribed to break down lactose. However, there is no lactose right now. You didn't need drink any milk, you didn't eat any cheese, no dairy products, none of that. There's no lactose. Operator has the repressor bind to it, blocking transcription from happening. RNA polymerase cannot bind to the promoter and transcribe the DNA. Okay? Now, let's go to scenario 2. I'm sorry, I meant to say RNA. It can't transcribe the genes. Okay? Scenario 2 is we have lactose present now. Lactose present. So now what happens here? There's lactose present. So you have all these little lactose here. They already came in because Lac Y allowed them in. Remember, Lac Y allows them to enter through the cell membrane. And now you have them coming into the cell, and now you have an isomer of lactose called allactose. All right, and what allolactose does is it binds to that repressor, so that repressor cannot fit on the operator. It acts as an inducer. Okay, remember, transcription here is usually turned off but needs to be turned on when lactose is present. So now we agreed that this is all our lactose. Here comes our allolactose, which is the isomer of lactose. And now we have this repressor trying to bind to the operator. But what's going to happen is this allolactose is going to act as an inducer. It changes its configuration. It's kind of like, think of it as a puzzle piece, There's another puzzle piece that fits here. And it blocks it from going into another one. Yeah, bad example, but I think you guys get the point. This allolactose blocks that repressor from binding onto the operator. So what's going to happen is this repressor is going to retract. It's going to go away. It's like, hey, dang, this allolactose, is blocking me from going on to the operator. So now what's going to happen is, now our boy RNA polymerase is able to transcribe all of these genes. Why? Because that repressor's out of the way now. So we can do anything we want. We're gonna transcribe all these genes so we could break down that lactose. So that's scenario 2. Okay, so those are the main two for the breaking down lactose. Now let's go into the next one. Now what happens when there is no glucose? Because remember, actually I forgot to mention this. When lactose is broken down into simpler sugars, it prefers to be broken down into glucose, right? So now we're going to talk about when there's no glucose. Now, if there's no glucose, what's going to happen? Let's start with there being lactose. There's lactose present in the cell. We know that the repressor is going to be blocked off because of our little isomer allolactose. And then since that's blocked, it's going to retract. It's going to go away. It's like, hey, I can't even bind to the operator. And we know that RNA polymerase is going to come in, transcribe these genes. Now this is where the capside comes in. Since there's no glucose, remember I said that the capside is a transcriptional activator. And what the cap stands for is catabolite activator protein. And we know that activator proteins, usually what they do is speed up a reaction. And that's exactly what the capside is going to do. Okay. Well, there's another thing that's involved with the capside. That's also where cyclic AMP binds to, which is a derivative. Actually, let me delete all of this to make room. Cyclic AMP, which is a derivative of ATP. Okay. And that's where it binds to the capside. So the capside is just telling the cyclic AMP where to go. So remember, we have RNA polymerase, and now we have the cyclic AMP here. Let me make that in white. Cyclic AMP that comes here, and that speeds up. More transcription happens. Way more, because remember, there's no glucose. So let's put this here. Low glucose or no glucose. High cyclic AMP. Wrote that big for you. Okay, and now for the last scenario. For the last scenario, we have glucose when there is glucose and there is lactose. Oops, let me write that over. Now we should be pros at this now. We know that there's lactose. There's lactose present in the cell. we know that we'll have this repressor here. And we know that it's going to get blocked by that isomer allolactose because there's lactose present. And RNA polymerase needs to come in and transcribe these genes. But now there's lactose involved. In the previous example, there was no lactose, and we need that cyclic AMP to really transcribe all of the genes. Now, since there is glucose, that means that cyclic AMP will not bind to the cap site. It's not needed. There's glucose there. So there's going to be very little transcription, or an average amount of transcription, not a lot, because there's already glucose present. Okay? So there's nothing that's going to bind there at all. So in this case, we have low or no glucose equals high. Oh, sorry. Actually, it's high glucose equals low cyclic AMP, or none at all. Okay, and that's just a brief overview of the Lac Operon. And just remember, you have these specialized genes that help break down lactose. The Lac Operon is a system that is usually turned off and must be turned on. And the reason being is there's no lactose. It's not going to waste its energy trying to transcribe these genes if it's not there. But if it is there, you're going to have an isomer that comes in called allolactose, blocks the repressor, so RNA polymerase can transcribe those genes. And then remember, lactose prefers to be broken down into glucose, a simpler sugar. When there is glucose involved, there'll be low cyclic AMP. However, if there is no glucose, you need cyclic AMP at that cap site to help the RNA polymerase make more transcription, so you can break down that lactose into glucose. Okay, and that concludes this video. Thank you guys. Leave a like, comment, subscribe. If you have any questions, you can email me or leave a comment down below. Try and grow this channel. I'm fairly new to this, so I'm trying to get better and better, help you guys get a better grade. So remember, just ask any questions. If you notice something, please ask. Thank you guys, and I'll see you next video.